Our next speaker is François Le Mauff. And François is currently a research associate in the lab of Dr. Don Sheppard at the University of McGill, at the Research Institute there. And François got his PhD from the University of Rouen in France, where he was studying protein glycosylation in microalgae and plants. And he's made the transition into the world of fungi. So François, without further ado, the floor is yours.
Thanks, Ryan, for this nice introduction. And I'm really happy to be here today from my home to be presenting this part of a nice collaboration that our lab shares with the lab of Linoel at SickKids. And today I'm going to speak more particularly about the result that we recently published in Nature Communication about AGD3. But before telling you a lot more about these proteins and why should we care about, let's put everything back in context and come back to the start. And everything starts with the Aspergilli. There is more than 250 species of Aspergilli that exist all around the globe. From the most desertic place to the Arctic circles, you'll find Aspergilli there, meaning that every single human on Earth is breathing spore of this fungi. The only thing is that when the spores reach the lungs of people suffering from an immunocompromised system or have impaired lung function, then those spores will feel really warm and cozy in there, start to germinate, grow, and provoke this lethal disease that we call an aspergillosis. And even though more than 250 species of Aspergillus exist around the globe, only one is provoking more than 80% of the cases of aspergillosis that we see. And this one is Aspergillus fumigatus, the fungus that I'm going to speak about today. So what is this fungus doing that the over or clearly not doing that's giving this great advantage and kill people? Well, when we look at the biofilm of this fungi under SEM, as you can see here with this, for example, including A. fumigatus, of course, you can see that A. fumigatus is making a lot of this white filamentous substance that is secreting all over the place and covering itself with, and the over fungi are really shy of making it. This substance is a polysaccharide. It's the one that we call Galactosaminogalactan, or GAG for short, and it's made of galactose and N-acetylgalactosamine. So how does Aspergillus fumigatus does it? This is thanks to these 5 enzymes that we see right here. And for the sake of time, I will not describe the full synthesis pathway. I will just attract your attention to this topmost enzyme right here, AGD3. Everything started when we looked at the different enzymes and mutated them one by one. When comes the turn of AGD3, we observed that when we made this mutant, the delta AGD3, the polymer was still able to be secreted, as you can see on the graph, but it was not processed anymore. The GalNAc residue stayed under GalNAc. There was no more galactosamine. And when we look at the fungus itself, we observed that delta AGD3 strain was not able to coat himself with this with the GAG that we saw for the wild type. The IP were totally naked in the same fashion that all the other Aspergilli are doing, the one that do not kill people. And that this fungus were not able to form an adherent biofilm at all. So we had here in AGD3 a perfect therapeutic target to kind of transform this Aspergillus fumigatus that covers himself with this polysaccharide and offers him the chance to kill people to this kind of IP, which is pretty innocuous as the other Aspergilli does. So we went straight on this enzyme and we had different projects around it, looking at what does it do in life, exactly what does it look like, and look for inhibitors that are able to inhibit the function of this protein. For the sake of time today, I'll not go on the inhibitor story. I'll stay on the structure and function of AGD3 to know more about this enzyme, because it's such an unusual enzyme, as you're going to be able to see. So if we look at AGD3 structure, you're going to be able to find this multi-domain structure that we have right here. And we were successful in crystallizing, so the part of the enzyme that you see right here, recombinantly expressed in yeast. And as you can see, we have kind of two parts in AGD3 by splitting in there. We have on the left part side, we have this alpha, beta, alpha fold that kind of form a sandwich with the beta-rich domain at the back in blue. And interestingly, none of the prediction software were able to match a function to this kind of structure. So we have no idea of what this part of a protein is doing. On the other side, on the pink part, we have this carbohydrate esterase domain. AGD3 is predicted to be a carbohydrate esterase 4. And here we kind of see that there is something a bit fishy in there. Usually the C4 have a domain that is less than 300 amino acid long. If we do a quick math right here, we can see that we are flirting with relatively 400 amino acid long sequence. So there is something going on that Natalie and I went after, aligning the sequence of AGD3 to the sequence of classical CE4 enzyme, such as IKB and PGAB that target PNAG, or PGDA and PDA. And we looked at what is making a classical CE4. The answer relies here in those 5 domains. Those 5 catalytic domain, MT1 and MT2, are mostly in charge of the coordination of the metal ion within the catalytic site using those 2 aspartic acids and those 2 histidine right there. We find them in AGD3. That seems great. It's not a good way to be a CE4. The only difference that we find there, it's right here. As you can see, in a classical CE4, those 2 domains are distanced by 50 amino acids. But when we looked at AGD3, we are more than doubling that, 130 amino acids. So what's going on? When we looked at the structure of the enzyme, so comparing AGD3 on the left to PGDA, for example, on the right, this addition, this 128 amino acids were highlighted here in orange with this insertion after the beta-2 strands, which is totally absent in the PGDA enzyme. Most of the amino acids that are in there are acidic residues, and they form these 3 little helices that we can see. And by doing so, they are deepening and putting a really high electronegative surface to this protein and contribute a lot to the formation of the binding cleft of the enzyme to the polysaccharide. If we go back now on the rest of the catalytic domain and see how AGD3 is performing, MT3 to MT5 contain the amino acids that are directly in charge of the DNA acetylation. And the most important residue in there is the acid residues that are initiating the reaction of DNA acetylation, this aspartic acid right in the MT4. But as you could see with the comparison of the elements, AGD3 doesn't have this residue. Though, when we look at the structure of the catalytic site, instead of the aspartic acids that we found in PGDA or in PDA, AGD3 has an ordered molecule of water right there. And it's not the only enzyme that has this molecule of water. Upon looking at literature, we were also able to find that IKB and PGAB has a molecule of water right in there, and we believe that it's actually replacing the aspartic acid to initiate the reaction of DNA acetylation. Of course, we verified the involvement of every single amino acid that is right there into the function of AGD3 using a mutagenesis approach and or ELISA to check the function of AGD3 through the stickiness of the products. I can go back to this unusual essay at the end if you're interested. And so following the same color code right there to the one that we have on top, you can see that as soon as we are mutating one of the amino acids that I spoke to you earlier, the function of AGD3 was totally abrogated, which allowed us to go back and to validate this reaction that we see right here, where we have the amino acids in charge, so the MT1 and MT2 in charge of coordinating the zinc metal and the water molecule. Then we have this water molecule that is attacking the acetate group from the GalNAc to form this intermediate of reaction right here, leading ultimately to separation of the acid acetic taken in charge by the zinc from the galactosamine that remain on the polymer. So This way to deacetylate is not mandated under the CE4 family. And when we submitted this, this paper to Kazim, they told us that IgG3 is actually a brand new carbohydrate esterase, and a family was created just for it, the CE18, a first creation since 2008. So quite an unusual enzyme that we have here. But the story is not finished. What's happening on the other side of the protein, this domain, this kind of subunit there, that we have absolutely no clue what it's for in life. We had the first idea of what it was doing when we were studying the function of IgG3 itself, when we were looking at how is it deacetylating. When we were kind of unlucky or not prepared to have pure oligosaccharide of GalNAc, we worked with purified oligosaccharide from the biofilm of Aspergillus fumigatus itself. So we degraded this biofilm, extracted the oligosaccharide from GAG, and got a mix from the degree of polymerization 5 to 16 that we observed through time using two parameters right here. So in the y-axis, you see the percentage of fully acetylated oligosaccharide, aka the rates of the first deacetylation event. And then we also reported the average number of deacetylation event through the color that you see on the graph. And when we get all of that together, you can see that there is a clear trend in the way IgG3 is processing the oligosaccharides. When the residues or when the polymers, the substrates are quite small, like 5 to 7, IgG3 is barely touching them. It's like not really deacetylate anything. But as soon as we reach 8, so this line right here, you can see that there is a drastic change in the way IgG3 was processing the sugar, managing to completely deacetylate it, and more we added length to the residue, faster IgG3 was able to go through them, reaching just a deacetylation in a matter of minutes when we were above 13 mer. And you can see also that more the polymer was long, more deacetylation was possible, since on our 16 mer at the end of a reaction that we observed there, the 16 mer become 9 times deacetylated right there. So we had a pretty good clue that IgG3 will really prefer the long oligomers. And when we looked at the structure of the protein, we have a really long protein with a really long cleft. Interestingly enough, when we looked at the structure on top of this, we see that the cleft that we have right here where the polysaccharide bind in the carbohydrate esterase is perfectly aligned with another cleft that is in this module that we don't know anything about. So it seems to fall in favor of this affinity increased by the long polymers. In addition, when we looked at the distance between the catalytic site and the entrance of this unknown module, we can see that it is about 33 angstrom. 33 angstrom match roughly with 8 residues of GalNAc link in alpha-1,4, such as in GAG, and this 8, this line right here, was when we saw the sudden change in the way IgG3 was processing the oligosaccharide. So we were here on a really good track to find that this module that we don't know anything about is actually a carbohydrate binding module that helped IgG3 to process the polysaccharide, which once again is quite unusual since normally carbohydrate esterase do not have a carbohydrate binding module that help them. We of course validated this using our favorite approach of mutagenesis and our ELISA to measure the activity of the enzyme. And we started by mutating residues that are at the far end of this module right there. So this aspartic acid in position 242. And you can see that we have absolutely no change in the activity of this enzyme. But as soon as we go inside this, this carbohydrate binding module targeting this tryptophan in 292, we lost 1 log in activity. And if we go straight at the entrance of this module with the tryptophan in 344, then this is where we had our most dramatic effect, losing 2 log in the activity of the enzyme. But this is just not a matter of activity that can overtake with time. For example, the trauma that we inflicted to the protein with this mutation is a lot deeper. when we looked at MSMS of the products to see where is the deacetylation performed by AGD3, we could see that when we use the wild-type enzyme, this one was constantly and systematically targeting the first residue on the non-reducing end. As soon as we mutated the tryptophan in position 344, you could see that the enzyme was quite lost. We had a random deacetylation among the first 5 residues, clearly showing us that without these residues, without this module, the enzyme was not able to properly lock in the polymer within the carbohydrate esterase domain and to process it as it should be. So we had right here a really unusual enzyme with a brand new family of carbohydrate esterase on one side, a brand new carbohydrate binding module that normally carbohydrate esterase do not have. This is AGD3. And this enzyme, even though it's new, original, first time reported, it is not alone. When we use the sequence of AGD3 to look for similar protein in other organisms, hundreds and hundreds of species showed up, as you could see here with this phylogenetic tree. And this is not only fungi. So fungi are representing the majority. This is this part of the phylogenetic tree, but we also find zoomycetes, and we also find a lot of bacteria, gram-negative and gram-positive, meaning that all this second part of a project that I did not have time to speak about today, looking for inhibitors of these proteins or strategies to inhibit this protein, could also potentially work for all these other species and their biofilm if they make so or if need be. On this note, I would like to thank, of course, all the collaborators that participating in this paper that we recently published, with a really special thanks to Nathalie Bonfort. She ran all the structural part that I presented today, and our 2 PIs, of course, so Don Sheppard and Noel, all the members of my lab and the OWEL lab, our institution, our funding agency, and I'm not forgetting you. Thank you for your attention.